Welcome to Chemistry 351. I'm Professor Andrus. I'll be your instructor for this semester. This is the 8-day M class here. You can see the different TAs in your recitation times. Make sure you know which recitation you're supposed to go to on Thursdays. Here are all the TAs. This is the abbreviated syllabus. A full PDF of the full syllabus is posted on Learning Suite. Make sure you familiarize yourself with all the content on Learning Suite. There's a lot of materials there, including the ebook, which is Organic Chemistry with Biological Topics. You can see here the 6th edition. We'll be covering these chapters 1 through 12, A, B, and C, and chapter 21. It's by Smith. Also included in the ebook is a study guide, a solution manual. That's included with your ebook purchase, which is the default mode. You don't need to do anything else. You'll be automatically billed for that book after the add drop deadline. If you want to opt out and get the book somewhere else, you need to do that by the add drop deadline, which is, I think, January 9th or so. I'll talk about that in class if you want to opt out. But by far the best approach to the textbook is the ebook. Just through Red Shelf, there's a button on top of the Learning Suite page where you just click on it and see the full content of the book and the solution manual. Molecular models are also recommended. They're available in the bookstore or in the Nichols building in the chemistry stockroom. You can get them there. Yes, we will do homework that's assigned here. One will be assigned for each chapter. It's 7 or 8 problems. You do them by hand on a sheet of paper and then upload that file to Learning Suite. There'll be an active button there you'll see under the assignments, and you just upload it by the deadline there. Make sure it's not an HEIC file right out of your iPhone. You need to convert it first to a PDF or a GIF to be compatible with Learning Suite. So make sure you do that. Homework book challenge is another thing you can do. It's your solutions to all of the even problems located at the end of each chapter. It's not graded. We use it as a borderline grade boost thing, and it's a way for you to get ready for exams and to be prepared for the final. It's due the day of the final, April 15th. There'll be a box outside my office. We'll talk more about that too. We'll also have quizzes. Some will be pop quizzes that are unannounced in class that you'll just do by hand a few minutes in class. Or we'll have some take-home quizzes that'll be posted on Learning Suite, and you'll see in the schedule how we'll do that. Again, you print those off, do it by hand, and then upload that file to Learning Suite by the deadline there. There are 4 midterm exams that are in the Testing Center closed book, covering 3 or 4 chapters at a time, 100 points each, and then the final is worth 200 points at the end at the regular scheduled time, which is, I think, April 15th. Yeah, Friday, April 15th for us, 11 a.m. right in our classroom, W. 112 there. There are also worksheet problems posted for each chapter. They're harder problems that we've given on older exams, and you can see those. They're not assigned or collected or anything. We use them in recitation or for you to just see how some of the harder problems might be. Organic chemistry. So here is the homework problems that are actually assigned. So homework problem 1 is for chapter 1, and it's 41, 45, etc., whatever. Those are the assigned ones. You have to show your work for that. Don't just copy down the solution manual. It's not just busy work. Make sure you show your work and understand those problems, and then upload that file to Learning Suite by the assigned time. So you see there, for all the chapters are listed there. They're also under Learning Suite, the syllabus and whatever. You can see the PDF of it. So these problems are available different places. But yeah, and the due dates you'll see on the schedule here in a second. Yeah, and I mentioned the file type there. Let's see, grades. Yeah, the 4 exams and then the final homework. We'll drop 2 of those, drop 1 of the quizzes, and those might be approximate. I'm not sure how many quizzes we'll do there. But the grade cutoffs, I use natural grade cutoffs where points group together. You guys create the curve there, approximately 20%, 30% A's and B's, and that can vary depending on how the points group together, whatever. But yeah, be familiar with Learning Suite, and of course all the materials there, the handouts and different things. We also have a display case west in the hallway there, down from W112, and your graded exams will be handed back in slots there. Make sure you pick them up soon. And any pop quizzes that we do in class, those will be handed back. But the ones on Learning Suite, the take-home quizzes, you'll have to look at the posted key to see how he graded that. So we'll not collect those and grade those by hand. So let's see. Yeah, you must attend class. Very important for you to come and stay ahead of me in the reading assignments you'll see on the schedule here. You know, come prepared, take notes, and ask good questions. Understand all the COVID things. The main things is we're still wearing masks in class, and the other issues here. So here's the schedule, quite detailed, I think, each chapter and the sections there and the topics you see going by. We'll start with structure and bonding. That's the first important thing. Yeah, it relates to what you've learned in general chemistry. That's very important. We'll get into reactivity quite soon with acid-base chemistry, and then unique things to OChem, functional groups and alkanes. And that's our first section. Chapters 1 through 4 will be exam 1. So you see we meet in class 3 times a week, 3 hours, so 3 credit hour class. The recommendation for study time for our department is 3 to 4 hours studying outside of class for every hour spent in class. So yeah, that's, you know, 9 to 12 hours or so every week you need to be working on material for this class. And you see assignments. We have something almost every day, or a couple each week anyway. So homework 1's due after we finish, you see chapter 1, and that button will be on Learning Suite. You'll just upload that file after you scan it or take a picture of it, whatever, of your work. And I like doing the work by hand. The electronic homework is difficult to work with. It's cumbersome, so we just go with the just taking a picture, scanning it, your hand done work. And then quiz 1 probably be take home. We'll have you upload that. That'll be due next Monday. Yeah, and you see the other assignments going by. Homework and quiz assignments are listed here. And of course the exam details are here. It's over the weekend, so Friday through Monday, January 28th through the 31st in the testing center. Closed book, of course, but you can use scratch paper and handheld models. We'll talk about that. And there's a data sheet on the exam, so you'll see some of the tabular data and some of the numbers you don't need to memorize, but you need to know how to use that. So you'll see what info will be available to you on the exam there. And then we'll get into stereoisomers, enantiomers, and stereochemistry, details of that, of course, unique to OChem. And then reactions. Here in the second section, we're doing reactions with alkyl halides, SN1, SN2 reactions, E2 reactions, eliminations, substitution reactions with different substrates and different types of nucleophiles and reactants. And that's really the second level of understanding in OChem. The first level is structure and bonding. The next thing's reactivity. What reagents and what substrates go together to form what type of products. Okay, and then the mechanisms, electron pushing, we'll get into that also. That's the third level of understanding there. Yeah, then we'll get into alcohols. Let's see, alkenes, alkynes, dienes. These are different types of functional groups in OChem. So they're grouped together that way with their respective reactions that are typical for them. There'll be 5 or 6 new reactions for each chapter. You'll have to know those. You'll have to know the structure and the reagents and the products that are generated from those different combinations there. Let's see, that's up to exam 3. Then exam 4, we have some spectroscopy here, mass spectrometry, NMR spectroscopy, and then a chapter on radicals. And yeah, that's the end there. And then we'll have a comprehensive review, which will be an ACS standardized final, American Chemical Society final. That'll be 70 multiple choice questions. My exams, midterm exams, will be part multiple choice and part write-on. So those long answers are a little more detailed. You have to write out some things, write out some products or some mechanisms there for part of the content of the midterm exam. But the final here will be all multiple choice. We'll prepare you for that, and we'll talk about that. But yeah, there's more detail in the homework assignments. I think we've already talked about that, and there they are, the exact numbers again. These are always at the end of the chapter problems there. These are the assigned ones for each chapter as they're due along here. Okay, how to succeed, how to survive. You all want an A, and I'll give you guys all A's if you're, getting the right number of points and doing well on exams. Of course, I'd give everybody A's if you were in that point range at the end. So how do you do it? Well, there's no mystery here. Organic chemistry takes time to learn. You have to read carefully, pay attention in class, and work enough with the problems. I think working the problems is kind of the key. That's where you develop some skills and some confidence in understanding the material and being able to work those problems. You understand issues of reactivity and selectivity, mechanisms, electron pushing, those things all begin to go together. So yeah, getting an A in OChem is easier than putting a camel through the eye of a needle here. The reference to the Bible there, impossible to get a rich man in heaven. No, it is possible to get an A in OChem here. But don't freak out, okay? Do these things and be careful, working the problems, staying ahead of us, attend reviews, recitation, office hours, the help room, we'll talk about that. There's an organic chemistry help room, W151, where TAs are available at all hours of the day. Participate in a study group. We'll talk about some group work and do some group activities in class from time to time. Make outlines, use flashcards, use those models if you have a hard time seeing things in 3 dimensions there. But why is organic chemistry important? So first day of class we'll talk about that. Why carbon? Why organic chemistry? You've probably heard about it from Gen Chem, but we need to get into the specifics now. Of course, developing new materials, new products, affluency, lifestyle, all these things are influenced by these new products that have been developed by chemists and engineers to make these different new materials. OChem is also important to satisfy curiosity, to know how everything works, how to make new materials, how everything works in biology and life systems. Cleaning up messes and different things are also an area that organic chemists work on. So there's a lot of areas there. Science is always a matter of size. They're very large here, where gravity, solar system, the universe, the earth, and plants and animals, that's what we're familiar with energy-wise, this force of gravity. Electrical forces we feel and see on smaller scale, microorganisms and cells, but these are still very large compared to actual molecules, which are on the nanometer scale, the angstrom level scale. But these small things, these molecules, this is an anti-cancer compound, Taxol, very cytotoxic. This kills cells at very low concentration. So something much, much smaller than a microorganism or a cell can be highly affected by something very small like a molecule here. And then the super small, the subatomic particles, we'll mention protons and neutrons and electrons, of course, from time to time. That has to do with bonding. This is really the domain of physicists here with the nuclear stuff. We're interested in localized electrical effects. Bonding is an electrical effect, right? The mutual attraction of electrons between the positive nuclei and atoms. So how those are held together to make molecules is what organic chemistry is dealing with. Structures. So yeah, why carbon? We'll talk about this the first day. Hopefully you're already thinking about this. Why a whole year studying carbon compounds? Why are you in this class? Why is your major program specifying that you need to take Chem 351 and 352? Most people go on to take 352. We use the same ebook, so you'll only be charged the one time for it, so don't worry about that. But why is carbon unique compared to the other elements, right? There's 100 other elements in the periodic table. Carbon is unique. What? 4 valence electrons. We'll have to review that. When it forms 4 bonds, you see, sharing those electrons with other 4 other atoms, it has what? 8 electrons now around that atom. That satisfies what? The octet rule. We'll have to talk about that. You see, oxygen, when it bonds, leaves behind lone pairs. So does nitrogen. Hydrogen just has 1 valence electron, so that's kind of boring. But we talk about hydrogen being bonded to carbon. Most of the structures will see a lot of carbon-hydrogen bonds. Carbon's unique there because it can adopt different shapes. If it's tetravalent, yeah, 4 bonds here. But if it's trivalent, bonding to 3 things, there's often a double bond left behind. That's sp2 hybridization versus sp3. Here we're mixing and matching the atomic orbitals, the s orbitals and the p atomic orbitals. If we mix them all together here and have 4 equivalent bonds here, we have a 3-dimensional shape, a tetrahedron. But if we have trigonal bonding with a double bond somewhere, they're all in a plane. This is a 2-dimensional object. And if we have a digonal structure with a triple bond, that's sp hybridization. We'll talk about this in chapter 1, but that's the unique thing about carbon, having these different shapes and possibilities here for bonding. A lot of the important things about structure and reactivity will come out of this understanding about valency and the bonding there. So yeah, just to give you a little intro here to different types of structures, because of that tetrahedral bonding, the 4 valence electrons, carbon can form extended chain-like structures. It can form ring-like structures. Different conformations of things like cyclohexane are important. We'll look at chair cyclohexane. Functional groups like alkenes, if there's a double bond to another carbon there, it's flat, trigonal planar. If it's double bonded to an oxygen, that's a trigonal planar carbonyl group. And then a collection of functional groups can come together, a benzene ring, an amide, another amide, a sulfide, and a carboxylic acid held together in this arrangement. That's penicillin. Some of you have probably taken that drug, but that's a 3-dimensional object held together with these bonding properties of carbon and the other atoms in the functional groups. And you know, it's important for making all these different materials that go into a lot of things that we enjoy in an affluent society. A lot of these are building materials that make different things like polymers and drugs and building materials and materials with different magnetic and electronic properties. A lot of these come out of petroleum, where initially we have a hydrocarbon material, long chain, nonpolar alkane material that's cracked to ethylene, which is a gas, and that can be easily converted to polyethylene and all these other different materials here. We'll learn a lot about those types of reactions and those materials. Like the alkene polymers, we'll see that in chapter 21. Cellulose and the sugar polymers we'll see in Chem 352. Fuel materials, we'll talk about isomers of these, and you see some naming issues here. Isooctane is a common name. 2,2,4-trimethylpentane is the systematic IUPAC name. So knowing the name, that relates to the structure and why that's an important fuel. We'll talk about that from kind of an engineering point of view. Here's some conducting polymers, polyacetylenes, reflective. It has a metal-like appearance to it. Polyanilines are in display materials, light-emitting diodes, different display liquid crystal materials that you see in display screens and other things. They have unique magnetic and electronic properties. Dyes and sensors have different colors associated with them, can respond to light and change their shape and appearance, and can sense different metals, different things. Yeah, I don't need to give you a lot of details here, but flavors and fragrances, maybe you can see a favorite one there, I don't know. Herbicides and pesticides, and of course drugs. We'll point out some of these as they relate to the different principles we'll cover in class. Chiral molecules, stereocenters, we'll talk about enantiomers in detail. Tetrahedral carbon has some unique aspects to it, some properties that if you have 4 different groups on that carbon right here, you can have different stereoisomeric variations, the S and the R enantiomers, and they have different properties, different conditions here, different tastes, different flavors, different activities in cells and as drugs, whatever. Okay, so yeah, a lot of you will be going on after Chem 352 to Chem 481, biochemistry. And these are all, of course, organic compounds, amino acids making up proteins in living systems. lipids, the barriers, the cell walls and membranes, carbohydrates, the energy molecules, and some other things for structure and bonding there. And then DNA, we see the central sugar here, the deoxyribose in DNA. And then RNA has a hydroxyl there, the ribose molecule. But then the base pairs and the genetic code, you'll get into that later in 352, but the basic structures and functional groups will be covering a lot of it in this class here. Proteins, here's a few of the amino acids. There's 20 normally that go into proteins. They link up and form polyamide structures here, so you can see some of those, how those relate. Structures of proteins, they fold up to form their active form, so you can see kind of a hierarchy of understanding the basic chem, how they link together, and then folding up, and then the function in biological systems. So you should see a progression in the ideas presented in some of your classes. Steroids, yeah, you're probably familiar with some of the hormones, steroids. These tetracyclic structures here all come from cholesterol, which is a component in cell membranes. Cholesterol is not a hormone, but it's converted into the hormones, and we'll talk about that in the functional groups, how they relate there. Here, the vision process comes from vitamin A down here, which comes from the diet, comes from different plant foods that we eat there. Carrots, for example, are high in carotene, which leads to vitamin A in the body when it's metabolized. In the eyeball, it's incorporated into opsin, rhodopsin, which is in the back of the eyeball here. And this is how light is harvested and how vision works. Light of different frequencies comes into the eyeball. And there are different isoforms of this protein that has the rhodopsin, has the highly unsaturated polyene that comes from retinal again. And when light impacts there, it changes the conformation from cis to trans. We'll talk about that type of stereoisomerization and what cis and trans mean here. There's a change in the conformation then of a G-coupled protein that leads to some signals that go into the optical nerve and then into the brain where the vision is actually created in your mind, right? But the mechanism, the mechanics of how light is harvested is based on the isomerization of this rather simple organic compound, rhodopsin. Anyway, synthesis organic compounds are made in the lab and in industry. We follow these materials by different analytical techniques depending on the scale we're doing it on. And different techniques are used to purify different products and then characterize them. We'll be going over NMR and IRO and mass spec. So yeah, these three types of analytical tools we'll be covering in 351. A little on the history here. This goes back about 200 years now. Organic chemistry's been rolling along, but it's really only the last 50 years or so we have the modern analytical techniques. We're able to fully understand things theoretically at a deep level. Initially, they were just trying to make different materials like Wohler making urea synthetically and acetic acid. These are natural materials, but we can make them synthetically. William Perkin, a young man in Britain, was trying to make quinine to help cure malaria, but instead he made mauve, this intensely purple material. It stained his lab coat and was kind of a nuisance there. But he recognized if he changed the structure a little bit, he could make different colors. And you notice staining-wise, this could kill different microorganisms under a microscope. So that's how the drug industry kind of started from Perkin's accident here. But we'll talk about that a little bit maybe. And then to sweat, developing silica gel chromatography to purify different mixtures of materials. Anyway, here's more of a modern thing, Tamiflu. Maybe you've heard of that drug, the antiviral drug. Of course, the antiviral drugs active against COVID are very important nowadays, ritonavir and Paxlovid. And maybe you've heard about some of the antibody drugs that are out there to treat severe COVID people. This was developed to treat influenza, which different strains can be very dangerous to people, of course. If you take Tamiflu within the first 48 hours, you're cured of the disease. But it's made synthetically in a 12-step sequence here. You see the diene and the dienophile doing a Diels-Alder reaction. We'll actually cover that in a later chapter, and then sequential reactions to make the actual drug here, Tamiflu. It's a sialidase inhibitor, which prevents the virus from merging with host cells, endothelial cells, in the lungs and the brachial tubes, and that prevents the virus from infecting someone. So it's kind of a molecular monkey wrench, we say. It binds this enzyme that's needed for infectivity, and it prevents the virus from coming in and infecting them. And so, yeah, the synthesis of it is organic chemistry, of course. How it binds and how it interacts, the conformation, is organic chemistry. But how the protein works and how the virus infects different cells, of course, biochemistry. Of course, there are organic chemistry aspects to all issues along the way there. Glycolysis will study the conversion of glucose onto pyruvate and eventually onto CO2 and water. This is the basics of metabolism in Chem 352. But you'll see some of these functional groups, and we'll mention some of these reactions here in 351. We'll also do the citric acid cycle, which converts acetyl-CoA onto carbon dioxide and then makes the ATP, the energy molecule you've probably heard about in all cells that are needed there. So OChem is primarily organic compounds, different materials here, different applications that you can see relating to their structures, reactivity, and different applications in different areas of study there. Biological chemistry, the four main types of molecules. Well, five really if you count amino acids separately. They combine together to make proteins. Carbohydrates or sugars, DNA, the nucleic acid material, the heredity molecules, and then the lipids, the nonpolar materials, including the hormones anyway. Analytical chemistry, yeah, we'll do a little bit of about this mass spec and NMR that we'll see later on. So hopefully that gives you a little overview to know what you're up against. But yeah, so how are you going to do this? Here's how you look today, right? Starting the semester, you all look sharp. And if you keep doing these things and keep being consistent and diligent in your approach to studying for this class, you're going to stay looking sharp like this. But if you leave out these things, you're going to look trashed at the end of the semester. This is how I always look at the end of each semester, so don't worry about that. Kind of scary, right? But yeah, if you stop doing these things, you'll start to get behind and have problems as well. So here's organic chemistry. Here's your brain. Hopefully there's enough room there in your brain to fit some of this stuff in there. OChem on the brain. The first thing is structure, bonding, how these things come together. You know, 4 valence electrons, what does that mean? Well, 4 stable bonds, no lone pairs, no empty orbitals when it bonds. Yeah, that's going to be a happy, stable molecule. Huge variety, because they can have chains and rings and functional groups, whatever. But that's the hole in your brain that you're going to fill with that understanding. The next thing is reactivity. You take one compound and react it with a reagent and make a new compound. That's the magic of chemistry, right? Converting, say, ethylene into ethanol. Very different properties, very different compounds, but understanding that reactivity, you know, that's the next level of understanding. Like I said, there'll be, you know, 5 or 6 new reactions each chapter you'll need to keep track of there. The next level of understanding is mechanism or electron pushing. We'll take filled orbitals, or where the electrons start. Here's a pair of electrons, a lone pair maybe on a non-carbon compound, filling into an empty orbital on a substrate or something in a system there. It's this flow of electrons that leads to the conversion of a starting material into a new product that you'll have to think about. So these are the 3 levels of understanding there that we'll need to point out repeatedly and keep you focused there, keep it well organized and everything. Yeah, so go to Learning Suite, look around, look at all the content, click on the buttons, see the things, the syllabus and the schedule is there. And we'll see you Monday morning, yeah, January 3rd, 8 a.m., bright and early in W112. Very good. See you soon.